A new podcast from the University of Michigan's College of LSA. This is How to Science with Professor Monica Deuce.
Discovering that scientists are humans and not robots. Here's a protocol for listening to this podcast. Step 1, turn off the laser microscope. Step 2, turn up the volume on the speaker by your beaker. Step 3, start listening. Super pretty pictures on Twitter of the tiny, cute water animal called Daphnia is how I first heard of Meg Duffy. The big eyes and feathery antenna of tiny Daphnia made me think of an anime version of a crustacean. So naturally, since I cannot resist cute things, I clicked on Meg's profile. And I found out two things. First, she was a professor at the University of Michigan, and second, her office is actually a floor below mine. Meg writes an incredible blog called Dynamic Ecology, which is about science, science life, and Meg's passion for diversity and inclusion in science. Meg and I talked about how scientists are not robots and how our feelings for the beauty and mystery of nature move us through science and its tough times. Today with us is Meg Duffy, who's a professor at the University of Michigan.
Hi, thanks for having me. So just to introduce myself a little bit more, I'm an associate professor in the Department of Ecology and Evolutionary Biology here at Michigan.
So how did you get into science? What's your science story?
I think when I talk to students, they assume that I had this really direct path. And if you look at my CV, it looks like I had a really direct path to science, but it did not feel that way to me. So I come from a very non-academic family. My mother's a nurse. My father is retired now, but he was a New York City firefighter. And so everyone in my family has these very practical sorts of jobs. You know, it's a family of nurses and firemen and electrical line workers and transit authority workers and things like that. And so I knew I was interested in science. I really liked my science classes all along. And now when I look back, I can see there were things where I was like, oh yeah, we watched The Voyage of the Mimi and I loved it. And that was about oceanography. And so, oh yeah, it makes sense that
that's like high school? Or
that was like in elementary school.
Oh, okay.
We watched that, yeah. And so I can look back and say, maybe it makes sense that I became an aquatic ecologist, except there were at least 100 other paths that were equally likely. So I was always interested in science, but I also knew that I didn't want to be a doctor. And so I wasn't exactly sure what I wanted to do, and I didn't really know how to navigate college very well, so I was just kind of stumbling through. And I didn't know ecology was a thing until I got to college. I was raised in a very religious Catholic family, and officially Catholicism accepts evolution, except the version of Catholicism I grew up with didn't. So I was kind of late in coming to ecology and evolutionary biology as a field. But then I thought fish seemed kind of interesting. And so I started working in a lab based on this project on fish speciation. And then while I was doing that, I heard about this other project and I was like, oh, that is really cool. And I switched to working on that. And that project was on the most polluted lake in the U.S., which is Onondaga Lake, which is near Syracuse, New York.
Okay. And
it had literally tons of mercury added to it, along with a host of other industrial pollutants, raw sewage.
By humans.
By humans. So very heavily impacted by humans. It's a super fun site. They've spent over $1 billion trying to clean it up,
wow.
And you still can't swim in it or eat the fish from it. So it was a really heavily polluted lake. And the lab I worked in had tracked how the zooplankton communities changed over time. So zooplankton are the small animals that swim in lakes. They're important because they eat algae and fish eat them. And over the course of several or many studies, what this lab showed is that the native species of zooplankton all died during the heavy pollution period. They were replaced by these really weird species that shouldn't have been in a big lake. And then as the lake recovered, it switched back to the original species assemblage. And so my project was, there was one species that was there during the heavily polluted time that they couldn't identify. And I used molecular genetics to identify
of zooplankton.
it. Yep. And so that was my introduction to working on Daphnia, which are these tiny little shrimp-like creatures that live in lakes. And they're what I still work on today.
Oh, that's really cool. So really you've like me, you've stuck with the same model organism, you fell in love with it and you never left it.
Yeah, exactly. So it's funny. I went back to Cornell a few years ago to give a seminar hosted by my undergrad advisor. And I was like, well, you could argue that I haven't developed much intellectually since I was an undergrad. Or I like to think I found a good study system early and I stuck with it.
And it's really funny because the way I learned about you is because you started tweeting a bunch of Daphnia pictures.
Oh, yeah. So I think Daphnia are really cool. I like them a lot, though. We use them as a model system to study infectious diseases. So I say that I love Daphnia, but I also give them these really horrible, virulent parasites. So I don't know, maybe I don't love them as much as I think I do. But part of why I love them is they're really ecologically important. So as I said, they're this key link in these lake food webs. They eat algae, so they keep our lakes from turning into pea soup.
Oh, okay.
And then fish eat them. So they're a really key link in these food webs in lake ecosystems. But they're also just a really convenient study system. So there are billions of them out in a lake, so we can collect many of them and it has no impact on the population dynamics. There are different species, so that we can ask questions about how diversity influences disease processes. There's this really kind of funny aspect of the system that ends up being really convenient, which is that they're transparent. Mm-hmm. That helps because then we can see what parasites they're infected with. And we just look under a microscope and we can tell. Whereas people who work on African buffalo and they have to go to these really great lengths to track down the buffalo.
Wow.
And then they put on a long glove and they just reach on in to get the intestinal parasites. And we don't have to do that.
Sounds cleaner.
Yeah, we just look under the scope and we can see if, based on their coloration and based on what the cells look like inside it, whether they're infected and what they're infected by.
And they look lovely. They have this little antennae and eyes.
Yes.
And you should go to Megan's website. She has a lot of pictures of Daphnia there. So they look really lovely too.
They're really cute. I really like taking pictures of them. It takes a long time, but I think they can be really striking. So I have a student in my lab now who's getting these really neat results related to what the Daphnia eat. And it's so cool because you can see their gut. And so you can see when they're eating a blue-green algae, they have a blue-green gut. And when they're eating the green algae, they have this bright green gut. And so you can just see all of that in them. You can see their heart beating. You can see the peristalsis in their gut. So one of the things that kept me working on Daphnia is just Daphnia mostly reproduce asexually, but sometimes they reproduce sexually. And when they reproduce sexually, they produce these long-lived resting stages that are basically like a seed. So the way a seed is for a plant, these resting eggs are for Daphnia. And the next year, some of them hatch out, but not all of them. And so you end up with these genetic archives back in time. It's almost like tree rings, right, where you can sort of go back along the different layers. And as you go deeper in the sediment, you're going back further in time. And so people do something that's called resurrection ecology, where you can take genotypes that were formed 100 years ago and hatch them out in the lab and compare them to modern genotypes. And so you can look at how they've responded to toxicants. You can look at how they've responded to changes in algae. Are they better at dealing with them? And it's just a really amazing aspect of the system. It turns out it's very hard technically to do these studies, so there aren't a lot of them. Well, I was in love with Daphnia. I was also somewhat in love with Antarctica. And so I had this brief foray. Instead of having a second semester of my senior year in college, I graduated a semester early and I went to Antarctica. And I worked as a technician there. And I mostly filtered water. It was not at all exciting research in terms of what I was doing on a day-to-day basis, but it was in a really amazing place. And I loved it there, but I also realized I really like doing local field work. If you're in Antarctica and something breaks and you don't have a backup, good luck. See what you can do with some zip ties and duct tape, but that's it. And I also, I just, I wanted to get a dog and things like that. I just realized were going to be a lot harder if I was doing field work remotely. So I went to Michigan State for grad school then, and I returned to Daphnia, and I've worked on them since then.
And that's where you did your PhD work.
Yeah. So I was at Michigan State, but I lived at the Kellogg Biological Station, which is in West Michigan, sort of near Kalamazoo. So it's a pretty small community, which I really liked. Some people, it's a little too small for them. But yeah, it's a really nice place. They have really nice lab facilities, and it's right on this lake and close to a bunch of other lakes. So for someone who is interested in lakes and the organisms that live in them, it was a really great place to do my work.
How was grad school? Was it what you expected?
Was it what I expected? So it's interesting because I, in general, at this point would say I'm very goal oriented and focused on what I need to do to get where I want to go. But that may be developed during grad school to some extent. So I got to grad school. I started doing this project that was on hybridization in Daphnia. I thought that seemed interesting, sort of how there was this species pair where sometimes they seemed, they never interbred. And then in some lakes, the species boundary totally broke down. And so at first I thought I was going to try to figure that out. And I just wasn't that into that project. So I worked on that my first 2 years, and I was just just not feeling super into this. And around that time, I learned that my advisor had seen that sometimes there were these big disease outbreaks in lakes, and no one had ever looked at them. Almost no one had ever noticed that they happened. But my advisor had noticed it. And I started thinking more about what I could do. And thinking about this egg bank that I mentioned before, that's really amazing, right? If you could go back in time and you can look at how the resistance of the Daphnia changed over time. And if you could track the parasites in the sediment too, it was just seemed like a really amazing system to look at that in. And so it was really the summer before my third year of grad school, and then the first half of my third year in grad school, I started working on Daphnia and their parasites. I was like, all right, I'm making this switch. I'd been a little nervous about making the switch because I felt like, well, I've already invested 2 years in this other project. But I spoke to one of my committee members, and he was like, you're setting yourself up to work on this for a career. If it takes an extra year or two to get that right, that's totally worth it. And that was really good advice. And so I decided to make this switch. And I was doing this really intensive sampling. So in that first year, I wanted to see what parasites were there and how they were affecting the population dynamic. Did they matter? I decided to do this sort of multi-pronged approach, I guess, where I had a set of 6 lakes that I was sampling really intensively. So every 3 days I was going to them. That on its own, now when I look back, I think that should have been it. That would on its own was probably a reasonable project. But then I was trying to pair it with this bigger survey where I was sampling, 12 other lakes less intensively.
So how do you sample a lake? Because it's pretty big. Usually.
Yeah. So we go out in rowboat. We take a net, which is basically like glorified pantyhose, where there's a ring on the top and you tow it through the water, and there's a bucket at the bottom with mesh in it. And so the water goes through, but it catches. So we go out to the deep basin in the lake, and we do this whole water column tow where we lower the net down to the bottom and we tow it all the way up to the top. And that tells you generally about what's out there in the water, but it doesn't give you the spatial structure. And so there's another way you can sample where you lower this big plexiglass bucket and you lower it down, and then it has these 2 trapdoors that you can trigger to close. And then you've trapped that little, 20 liters or so of water in that spot. And then you can bring that up to the surface. And if you do that every meter or so, then you can see where the Daphnia are living in the water column. And we know that the Daphnia move day and night. So they tend to be deep during the day, and then they move up at night.
They're vampires.
Yeah. It's probably a combination of avoiding fish predation. So fish can see them during the day, so they have to go down deep to hide. Some of it might be the food is better down deep too. And some of it might be at night they can go up and their eggs can develop faster. So they're invertebrates, so the temperature influences their egg development time, and it's warmer when you're up higher in a lake. So they're probably migrating for different reasons, but we know they're migrating. And so in addition to doing these surveys of these lakes, I was also trying to quantify where they were in the water column. So I was going out and doing these samples where I was collecting samples every meter, and you do that during the day, and then you do it again at night. And with parasites in Daphnia, so I said Daphnia have all these wonderful advantages, and I love them. A disadvantage of the system is once you preserve your samples, you can't tell if they were infected anymore. So we have to count everything live. So we have to do all our counts within a day or so of collecting them. So I was doing these samples, though, and it was amazing. So I was finding there were these bright red Daphnia. I could see they were down deep in the water, and they were staying deep even at night. They weren't migrating up. And I thought, whoa, this is so cool. So I knew some Daphnia make hemoglobin, but there was a paper saying the species of Daphnia I work on does not make hemoglobin. And I knew that there was a parasite that turns Daphnia red. And so I was like, this is amazing. They're infected with this parasite. And I wasn't sure if it was like maybe they aren't moving up because they're infected and they like can't swim well or something like that. So I had all these hypotheses, and I was really exhausting myself trying to do all this different work to really figure this out. And at some point I thought, well, I want to know how virulent this parasite is. So I took some uninfected Daphnia and I put them each singly in a beaker. And then I did the same with some of these red Daphnia. And then I checked them every day. And I did that for a few days, and they were all living. And then a few weeks, and they were still all living. And at some point I was like, something isn't right here. And so I pulled a few of the ones from my infected treatment out and looked at them under the microscope, and they just looked perfectly healthy. It was really an aha moment. As I was looking, it was a bad aha moment. I was looking under the scope, and I was like, they were never infected. They make hemoglobin. When I looked at them, I was like, that's why they were deeper, because there's less oxygen down deep. That's why they were red and it went away. That's why none of them died. And so, as a reminder, this was after I had felt like maybe it's a little too late to switch to working on parasites. And so I was now most of the way through the field season in my third year in grad school, where I thought I was doing a thesis on parasites, and where I realized I had no data on parasites.
Wow.
And I just started crying right at the scope, and I got up and I walked outside. There was a bench next to the lake, and I just sat there and cried. And I was like, well, I'd seen an ad that morning saying that this person who does really amazing work on Daphnia was looking for a technician. And I was like, all right, that's it. I'm just going to quit grad school and go and be his technician. And that's just how it's going to be. And I was just like, all right, that's it. I'm done. And for a few days, I was just like, okay, this is what it's going to be. Mm-hmm. And my advisor had been at NSF that year as a rotating program officer, but he just happened to be back visiting right when I figured this out. And he was really amazing. And he had me over to his house for dinner, and he was like, I know it seems really bad now, but it will be okay. And in the end, I mean, obviously I was unlucky that all of that had happened, but I discovered it basically just in time to salvage that field season. There was just enough time to still collect enough data to do interesting work. But at that time, it just seemed hopeless. It was like, I'm roughly halfway through my grad program and I have no data, and there's just no way this is going to work. But it did. It ended up working out. And actually there was a point where, so it took several months before I could even think about those data. But at some point I was like, there was a really neat pattern there. I mean, there was this really striking pattern of where these things were living, and it was really different. And I realized that hemoglobin then was a marker for where they were living. So it actually meant I didn't have to exhaust myself doing these samples. I could just look at whether they were red, and if they were red, they were staying deep all the time, and if they were clear, they weren't. And it ended up being really neat. And so we started jokingly referring to it as thesis 2. I did end up writing that up, and it's a publication, and I think it's a reasonably interesting one. So migration behavior in Daphnia, what's controlling it? What are the impacts of it? So I think that's something that we'll probably end up following up on, this totally accidental study that made me almost quit grad school.
Yeah, that made you quit. And I feel like this outpour of emotion is something that perhaps we don't really portray well to the public, but it happens almost every day in science. Maybe not to like the same extent where you're like, that's it.
Yeah.
But so many other times I found myself crying because something goes horribly wrong, or something I've invested a lot of time doesn't work out. And I think it really shows you that there's a deep emotional connection that we have with science. And we do it because it's interesting and it might help people or the environment, but there's more to that. It's sort of like you feel so strongly in love with it, and it's almost like our unrequited love.
Well, and I think there's this message that to be a scientist, you have to be just completely rational, and that there's no room for emotion. And if you're emotional, you're not a good scientist. And yes, when I'm analyzing my data, I am trying to be as rational as I can. But I think it's not a good thing that we send this message that if you have emotions, that means you're not a good scientist. So I have a blog post. So I write for a blog, Dynamic Ecology, and I have a blog post that's called "There is Crying in Science." That's okay. And so I think it's something that's taken me a long time to really accept. But yeah, like I'm not a robot, and that's okay. That's actually a good thing.
I think that's great, and that's something I actually think about a lot. And as a graduate student, and really even as a postdoc, I didn't really have any women mentor, and there weren't many women in science. And so there was this idea that you had to be a robot and you have to be hyper rational and never show any sign of weakness and crying, but even just saying that things were tough were clearly signs of acute weakness, and maybe you were just not cut it. You weren't resilient enough. So part of it is science completely fills your life, and it's this really huge structure where you really have to go through a lot of hoops. There's like 4 walls for which to bounce around and get hurt or succeed. But at the same time, you do that all the time and you spend all your life and effort in it. So I think it's normal to have such emotional reaction to it. So where do you think this idea that scientists should be robot came from?
I don't really know. I mean, I just feel like it's one of these things that's really damaging, because it means that some people who potentially could contribute a lot to science get the message that maybe science isn't for them because they're emotional and you're not supposed to be emotional as a scientist. Like if they get that message, then I think it might make some people think it's not the right career path when actually they could be really great scientists.
Absolutely. And I also think that's one of the reasons why people don't trust scientists, because I honestly wouldn't trust somebody who doesn't have emotion.
Yeah.
There's a lot of studies that show that the inability to sense your own skin crawling actually puts you in dangerous situations. So clearly emotion is a really important component to keep us alive.
So I write for a blog that's aimed at other academics. And so I definitely feel like a really useful aspect of the blog is to write these sort of stories about how I almost quit science, or grad school at least. And we know that many of the readers of our blog are grad students and postdocs. And I think sending those messages that are like, yeah, I almost dropped out too. It's pretty common for students about halfway through grad school to have a point where they're not sure they can do it. And I think it made me interested in blogging so I could tell those stories more. Another reason I was interested in blogging, though, was for diversity-related reasons. And I really like having that platform to talk about those issues. The outreach work I do tends to be aimed at trying to broaden participation in science.
What made you first interested in doing it, or what alerted you that it was an issue that you really wanted to spend time and effort on? Clearly, that's a really important one, especially now, and Michigan's really involved with it. But you've been doing that for a while, even at your previous faculty position in Georgia, and you received some awards for it too, I think.
Yeah.
So what drew you into?
Well, I think some of it was being at Georgia Tech, which is in Midtown Atlanta. Our main field site was in Midtown Atlanta, and Atlanta is this wonderfully diverse city. And it just seemed like, of course, I want to talk about the research that we're doing and try to connect the research that we're doing with the people who live here. I don't know, it just seemed obvious. Like, of course, I would try to reach out to people in the Atlanta public school system. A lot of the work we did at Georgia Tech was through the Piedmont Park Conservancy. So Piedmont Park is in Midtown Atlanta. It has this lake in it. It's pretty big considering that it's in Midtown Atlanta. And so we used that as our field site. And we worked then with summer camps that were at the park. And we would have the kids go out on the dock and they would collect samples and then they would look at them under the microscope. And we would start out the activities this way. We'd ask them what's in the lake, and they'd say, trash. We're like, well, yeah, that's true. And they could usually get like fish and turtles, and they'd guess sharks. And then we'd be like, oh, no sharks in the lake. But they haven't been exposed to the other things that are in there. And at some level, I think that resonates to me because I wasn't exposed to ecology really as a kid. And so some of it is the stuff I would have really loved to know about as a kid that I didn't know about. I come from a very blue-collar family. And so at some level, I guess I just connect with people who in general have not been connected with academia, and trying to increase those connections just makes a lot of sense to me.
And I guess as an outsider, it seems that there is a connection between what you study, because you're studying an ecosystem, a lake, and the creatures that live in it, and the pollution, and how that influences each other, the lake, the creatures all together. But that's the way we think about humans, right?
Yep.
In the cities, and there's an environment, and it's sort of like, if I do this, can I enact a change that's positive?
Yeah, well, and I think also as an ecologist, I realize there are a lot of benefits to diversity.
Absolutely.
Systems are more productive when you have a more diverse system. That's true for a prairie. And I think that's true for science, too. And so I recently wrote an opinion piece that relates to this lake that I did my undergraduate research on. And I was thinking that it's interesting. One of the ways we know things are improving in that lake is the diversity is increasing in the lake. I was like, well, that's pretty parallel, with science. A sign that things are improving in science would be an increase in diversity in science. They will notice different things. They will think about different things. They'll approach questions from different angles. And that's good, right? Like if we're all trying to address the same questions from the same angle, that's not good for science or for anyone. But lately, what we've gotten really involved in is Wolverine Pathways, this really amazing program for middle and high school students from Ypsilanti and Southfield, and it's expanding into Detroit. And if they complete the whole program, so if they get through their senior year, and they get admitted to Michigan, they get a full tuition scholarship for all 4 years to Michigan. The students who are in it are coming from underserved communities. And so it just seems like such an amazing program. And so once I heard about it, I was like, I really want to be involved in it. So next year will be the first year there are seniors in the Wolverine Pathways program. And in their senior year, they'll have to do a capstone project. And the program doesn't have a STEM focus. So for some of them, their capstone project will be like writing a collection of poetry or something like that. But for many of them, it will probably have a quantitative component. But a lot of the motivation at this point is just to give them the skills they need to think critically about questions they care about. And so we're calling the activity Prove It. So they have to take a question they're interested in, and then find and access and work with data to come to some conclusion related to that question, and then they have to present it. It would be great if it was a question that related to the greater good in their communities, but if their question is Steph Curry the greatest basketball player ever, that's fine. I mean, they'd have to refine it to get to a question that they could address
use
with
data, I guess.
Yeah, but I think that process of coming up with a question they're interested in and then thinking about how to refine it into a question that you can address with data, like that on its own will be really useful. So they'll have to figure out how to take a question and turn it into something they can address. They'll have to learn how you access reliable sources of information. So some of this will just be information literacy. If you go to a website, what are things that would tell you that it's reliable or not? How do you find data on the web? Some of it will actually be working with it. Like, so how do you, in Excel, do a data analysis in a way where 6 months from now, you know what you did? So Wolverine Pathways is one partner in it. The other partner is this group, Software Carpentry, which is an international group that aims to train scientists in how to do reproducible data analyses. But one of the things that we like to say in software carpentry is that your primary collaborator is yourself 6 months from now, and your past self doesn't answer email. And so we want to get that idea across to these students that in 6 months, if they want to follow up on this for their senior capstone project, they're going to wish they took really good notes. Or maybe someone in the program next year will be interested in what they did, and they'll want to see how things have changed in the past year. And so they'll want to repeat what they did. And so over the course of the 2 weeks, we'll walk them through this different process of developing and refining a question and then working with data and then how to present data. So just one of my goals is that if we can train these high school students in how to do that, they'll both be more informed consumers as they just live their lives, but they'll also be like have a jump start on what some of the skills they'll need when they get to college.
Yeah, so critical thinking is not just a superpower, but it's a common thing everybody has.
Yeah. So we're going to hopefully help them develop those skills that they'll need when, if their uncle says something at Thanksgiving, they can be like, I'm not sure about that. And then go and answer that question.
So, thinking about what you're interested in science and in science communication, this idea of the ecosystem and how the individuals and the species that live there and the system as a whole influence each other. How do you feel that science and outreach has changed you as a person? What's the effect of the ecosystem on you?
That's interesting. Well, it's hard to know in some ways how science has affected me as a person because obviously science sort of reinforces some traits of mine and strengthens them, but they're ones that were there already. So I have 3 kids and I have a spreadsheet that has milestones for each of them, right? That's such a dorky science thing to do. But if I wasn't a scientist, I probably would still, I might still have a spreadsheet for them, right? So it's hard to know for some things like that how much of it is because I'm a scientist or how much I'm a scientist because I have those sorts of tendencies.
Or it's reinforcing those tendencies.
Yes, exactly.
For sure.
It's reinforcing that. I think as I progress in my career, I think I increasingly realize and think more about questions that are maybe more related to social sciences or history. And one of the things I think is really interesting, if I look back, it looks like I had this direct path to where I am now. But there were so many other paths that could have seemed in the end as direct, some of which are completely different, where I would be just as happy. I don't know. I just think it's kind of fun to think about all the different pathways I might have taken.
And I think that's quintessentially what's being a scientist. It's really all about that you're really interested in the world, which is made of humans and other animals and plants and everything. And you just really want to discover what it's like and what it's beautiful about it.
Yeah, and I've had some students who they're almost paralyzed by, they have a lot of different interests and they've received this message that there is one true path and they have to find their one true path that will fulfill them. And I feel like probably there are a lot of paths for a lot of people that they would be happy in. And maybe they would start down one path and then realize they're not happy in it and they can switch and take another path, and that's fine too. I say this to all the students, the undergrads who work in my lab. Like, if you join the lab and you decide this isn't what you're interested in, that is totally fine. That was not a waste of your time. Figuring out what you're not interested in is just as valuable as figuring out what you are interested in. But again, I think we tend to have these narratives that are, this person was destined to be whatever profession they're in since they were a child. And we maybe don't call enough attention to that there are a lot of different paths that people could take. And there are a lot of chance events that get us to where we are. Right. And that with a different roll of the dice, we would be in some other career path. And that's fine.
So if you're enjoying finding out about life beyond the beaker and discovering that scientists are not robots, but real people, help us spread the word about How to Science by leaving a review on Apple Podcast. Go on Twitter, #HowToScience. Tell people you like it. Tell us if you liked it. Tell us what you want to hear next. Thank you to LSA, the liberal arts college at the University of Michigan, for being the force behind the podcast and really doing all the work. Thank you to WCBN-FM, the University of Michigan's campus radio station, for letting us use their cozy, grungy space to record the podcast. Thank you to the Department of Molecular, Cellular, and Developmental Biology, where I spend most of my science time. Thank you to the Liz.
Find the How to Science podcast on Stitcher, Apple Podcasts, or lsa.umich.edu. That's lsa.umich.edu. Or wherever else sound is available on the internet. Visit weekly for new episodes in the series. Podcast theme music by Poddington Bear. How to Science with Professor Monica Deuce, who says,
I'm not a funny person, I'm only accidentally funny.